Well, good morning, everyone. I'm going to give it another minute or two. People are still coming in. And I'm glad to see that the founding manager of the Catskill Science Collaborative, Jamie Deppens, logged in. Hi, Jamie. Thank you for coming. I will introduce myself first. Up to 21. We'll give another second. I'm Josh Ginsberg. I oversee the Catskill Science Collaborative when I'm not president of Cary Institute of Ecosystem Studies. And it's been a real pleasure. Gary Lovett, who is the founder and really the brainchild who came up with the idea of the Catskill Science Collaborative, handed off to me about 6-9 months ago. So let me tell you a little bit about the history of the collaborative. The Catskill Environmental Research and Monitoring Program, CERM, has been going for over a decade. It is a wonderful, loosely organized, but very, very committed group of people who care about science in the Catskills. And every 2 years, well, it's going to be, I think, 4 years now because of the pandemic, we have held a seminar, a meeting, often at Bel Air Ski Center, to bring together those people who care about science in the Catskills. And we realized that there was a real need to provoke and amplify interest in science in the Catskills. The Adirondacks have always been sort of like the rich, and in here I use rich intellectually, the rich older brother of the Catskills. And while there are many people who care deeply about the Catskills, there wasn't really an organizational structure to promote and amplify work that is done in the Catskills with science. So after some conversations, Cary, working with the Department of Environmental Conservation, through whom we got the core grant from the Environmental Protection Fund, which they manage for the state. The grant gave us the opportunity to launch the Catskill Science Collaborative. And the collaborative does 3 things. The first thing we do is what we're going to talk about today. We fund young scientists and their mentors to do research in the Catskills. We do this for 2 reasons. The research projects are developed in coordination with DEC, the Department of Environmental Conservation of the state of New York, and DEP, the Department of Environmental Protection of the city of New York. As many of you know, New York has some of the best water in the world of any major city. It's part of why a friend of mine, Catherine Hines, called it the greenest city in the country. And that comes from a 100-year-old reservoir system in the Catskills. So DEP has a very strong interest in land management, particularly how it affects the water quality of those reservoirs. Anyway, we get topics and ideas from our partners and then work with our academic partners and their students to develop research projects. And we're going to be talking about 2 of them today, one on freshwater invasives and one on forest regeneration. We also are capturing their data and historical data in 2 databases, one of which is a set of data sets, which will be online and available for managers and others to use as they would like to understand the Catskills better. And then a bibliographic data set that is up to almost 1,000 references in a program called Zotero, which will be online as well. And that will give people access both to the science we fund, but even more importantly, to the science that has been done historically, including things like Mike Kudish's data sets on geographically tagged data sets on trees and forests and others on animals and birds. Mike is really one of the great experts and ecologists in the Catskills. And so getting his data up online is going to be a remarkable thing. We've started that project. The third leg of the stool for the Catskill Science Collaborative is also collaborative, and that's trying to do outreach to tell people about the Catskills and the way science informs management. We do that in real life in the old days, and we've been doing it virtually for a couple of years. That's in partnership with DEC and also the Catskill Center for Conservation and Development and the Catskill Mountain Keeper. And we're very pleased to be partnering with others because this really helps amplify our message. And my guess is if we can do it technologically, we will never do a only in real life event again. There'll always be hybrid because we're able to get a lot more people into these sessions by having them virtual. So that's the Catskill Science Collaborative. It is hosted by the Cary Institute of Ecosystem Studies in Millbrook, New York. The Carey Institute has a long history of working in the Catskills. Gary Lovett, Kathy Weathers, and many others have done research there for a couple decades. And so we are proud to host it. And that is part of what we do. We also do work not just on forest and freshwater, but on disease ecology and on urban ecology. The disease ecology work includes work on pandemic spillover, so we've been busy, and on the impacts of biodiversity on disease spillover and human health. So we've got lots going on at the Carey, but we are very proud to host the Catskill Science Collaborative. And with that, I will hand over to the manager of the CSC, Kira Peterson. Kira, all yours.
Thank you, Josh. I'm really grateful for everybody who's here. Some people have already started. You're welcome to say hello and introduce yourself, say where you're calling in from in the chat. I'm sure that would be nice for everyone to see. And as Josh said, I'm new to the Catskill Science Collaborative. I'm very excited to be here. And because we are going to be hearing about science on the theme of invasives today, we thought it would be very fitting to start off with a little bit of information on the Catskill Regional Invasive Species Partnership from John Thompson. So John, take it away.
Thanks. I'll grab my slides here and let me know when you can see them. Oops.
Looks great.
Can you see it? Great.
Mm-hmm.
Thank you. So I'm John Thompson. I'm the Catskill Regional Invasive Species Partnership Coordinator. I coordinate one of 8 partnerships for regional invasive species management in New York State. We are also primarily funded from the New York State Environmental Protection Fund. Our work here is to promote education, prevention, early detection, and control of invasive species to limit their impact on the ecosystems and economies of the Catskills. And I'll show you the area that we work in. We'll review some of the highlights from our current 5-year contract and talk a little bit about what we expect to do the next 5 years and how these projects are going to help us. So the CRISP region is shown on this map, outlined in red. It's an area of about 3.3 million acres. The Catskill blue line, which you're probably all familiar with, is here. That's about 700,000 acres. The yellow star that you see there is where the Catskill Center is located in Arkville. The Catskill Center for Conservation and Development is the host organization for CRISP. So the CRISP region includes all of Otsego, Schoharie, and Delaware counties, most of Greene County, most of Ulster County, most of Sullivan County, and a small part of Orange County near Port Jervis. We are working with a number of partners within this region, including the Catskill Science Collaborative, but a variety of partners who are concerned with invasive species, working in invasive species in different ways, whether it be education or control, interacting with the public in different ways. And some of the major landowners that we work with are New York State DC. They own over 400,000 acres in our region, and the New York City DEP manages over 150,000 acres in our region. So those are some of our primary partners. Just to give you some highlights from our current 5-year contract, which ends this year, we led 252 programs and events for nearly 24,000 participants, probably over 24,000 participants now. We conducted 106 trainings for 1,322 participants. Generally, we are working with people, showing them the early detection species that we want them to recognize, like spotted lanternfly that was on my first slide, or mile-a-minute that we have here. Those are species that we want to identify quickly and go out and respond rapidly to. We supported Cornell Cooperative Extension to do additional invasive species outreach to nearly 155,000 people in the last 5 years. We've surveyed well over 4,000 acres for invasive species, and we eradicated giant hogweed at 5 of our sites. And generally, we're working with private landowners to get access to do these surveys, to do our control. But we also, as I mentioned, we work on state property and also city property to a large degree. So looking forward, we're looking to expand our staff to have an aquatic invasive species team working with us, expanding our terrestrial invasive species staff to have a coordinator and also a strike team that are going out and doing control projects. And we'll continue to release requests for proposals to fund projects that meet CRIS priorities. So we're planning to do that next year and in the following years in our contract. And I'd like to mention that I've been really fortunate to work with the Catskill Science Collaborative Fellows. These 2 projects that we're going to hear about this morning are very important to the work that we do and the work that we're seeking to do in the next 5 years, including identifying priority water bodies to survey with these people shown on the screen, and understanding the interactions of invasive species and native species like deer, so that we can know what the best management actions might be going forward. So as I said, I'm very excited to hear these presentations today and the great work that they're doing. And just quickly, I just wanted to draw people's attention to a documentary about the New York State Invasive Species Program that premieres on the DC's YouTube channel on Monday, November 1st. So look for that, search for that on Google, and hopefully you can watch that and learn more about the great programs that are happening with invasive species management in New York State. So I'm going to stop sharing.
Yeah, thank you so much, John.
Thanks.
I'm going to welcome anyone also while the first group of fellows are presenting to write questions that you have in the chat, or save your questions for the end and write them in the chat, and then I will read them aloud and give our fellows a chance to answer you. So without further ado, I'm going to let Hani Mendoza and TJ McKiernan tell us about aquatic invasive species in the Catskills region. They're from Binghamton University. So welcome, Hani and TJ.
Hi, do you hear me okay?
Yep.
Okay, so good morning, everyone. My name is Hani Mendoza. I work together with Thomas and our advisor, Dr. Laura Pengalozzi from Binghamton University as well. Our project is about aquatic invasive species in the Catskill region. And I will start by explaining a little bit of background. As you know, when we are doing a project, we have to start by explaining what the concept we are going to talking about means. So we started by defining what an invasive species was. And an invasive species is an introduced species that threatens biodiversity or poses another problem. Invasive species are a tiny fraction of all introduced species, and it is important to recognize that introduced species and invasive species are not the same thing. An introduced species is one outside the habitat where it evolved to present form. And an invasive species, it's basically, as we said earlier, a species that poses some kind of threat. In New York State, about a third of all species are introduced, primarily from Europe, and they came under colonization. Of the thousands of introduced species in New York State, the Catskill Regional Invasive Species Partnership lists 44 aquatic plants and animals of concern, subdivided into 5 tiers of declining concern, from early detection to monitoring, which is tier number 5. It is important to know that very few introduced species are harmful to people or other organisms, and many are beneficial, opposed to what many people think. All introduced species are not harmful. I personally personally had the belief before doing this project that all introduced species were harmful. And this has opened my mind in many different ways, just because I realized that indeed many of the introduced species that we have today in the United States are very, very beneficial for us and for other animals and different kinds of species as well. For example, the red swamp crayfish, it's native to North America. It is now invasive in Europe, but serves as an abundant food resource for threatened water birds. As I said, many invasives have benefits, just like honeysuckle, autumn olive, oriental bittersweet, and garlic. Who would have thought that garlic was an invasive species and is widely used everywhere, but it is invasive in North America. Still may reduce biodiversity, but balanced against benefits. So even though garlic and such a like autumn olive, they're invasive, they still like, we have to balance if they are harmful or beneficial. But since they're more beneficial to us, we have to balance that. Native species can also be species of concern. There are many native species that even though they're native, they can also be invasive and become detrimental for many different ecosystems. For example, the microbe that causes Lyme disease, it is native, but it has gone out of control, we can say. We got this information from the Sage Handbook of Biogeography that was written by Professor Blumblur from Binghamton University. And then there rises the question, like, why are we only studying aquatic freshwater species only? And it's because the rapid growth in vegetation has become a problem for managed waterways, just like the Catskill Park. And then why are aquatic invasive species a particular issue in the Catskills? Well, as we may know already, the Catskill Park, it's basically the main source of drinking water for New York City and surrounding areas. The Catskill Park contains 4 reservoirs for New York City, as I said, and several others that are part of local water supply. Excess vegetation in water can become a problem for reservoirs, and that's the reason why vegetation is cleared from banks. When vegetation dies, it creates humic substances, yellow-brown matter, that primary form of organic carbon-containing compounds in drinking water. These humic substances react with chlorine, which is the main chemical used to, let's say, treat water. And when these humic substances react with chlorine, they produce bromine compounds, and these may increase cancer rates, which is concerning due to the fact that a lot of people, in fact, a little bit more than 10 million people depend on these water reservoirs for their everyday water needs. Yeah, as I said, chlorine is the most used water disinfectant, and it's because it's inexpensive. The resulting compounds together with chlorine and, humic substances, they are called trihalomethanes, which are, as we may know, they are detrimental for our environment. And these are regulated by the EPA under the disinfection by products rule. If New York City does not control vegetation in water, would have to build more water treatment plants, and this is going to be more expensive. For example, the Croton filtration plant just completed at $3.2 billion in 2015. And this is the reason why it is important for us to study why invasive aquatic species are detrimental to waterways, because we as humans depend on these waterways. And then we have, what questions are we trying to answer with our project? And first of all, we want to explain our area of study. First, we were only gonna study the Catskill Park, but as we know, there aren't many invasive species just in the Catskill Park, which is that blue area delineated in this map. Maybe because it's a protected area. So we decided to expand to all the crisp area. Yep, we don't have so much data in the Catskill Park, as I said. And then where are the risks of aquatic invasive species in the Catskill region? When first of all, we have recreational activities, including boating and fishing. So wherever we have like a lake or any waterway that is used for recreation purposes, we have a lot more of chances of having invasive species in that area. And we have set our dependent variable for the study, the location. So based on the location, there's gonna be many different things that we can assess from there. And then TJ is gonna go ahead and explain what independent variables may influence the location of invasive species. Thank you.
Thank you, Hani. So first, I'm just going to talk about some of the maps we've made to look at the distribution of invasive species in this CRISP region. So the first map we're looking at is a heat map within CRISP that shows the northern part of the CRISP region containing the most dense concentration of invasive species sightings. Oh, there we go. So a nearest neighbor function was used, which compares the distance between, say, one sighting of an invasive species and the next closest invasive species. And the higher concentration areas are denoted by the purple rings. These numbers represent invasive species count within the hydraulic unit codes. So for each of these little polygons you see on the map, there are smaller watersheds, because if we had divided this region into the watersheds that we know exist, we would be left with the Susquehanna, Delaware, Lower Hudson, and Mohawk, which would just be large areas for an analysis that wants to look at specific spots where invasives may be threatening the ecosystem. So we divided it into the HUC 10 boundaries, which are much smaller, and they're easier to identify where the invasive species are concentrated. So like Hani said, there was an absence of clusters within the blue line of the Catskill Park, except towards the western part of the park. And we know that that's probably because it's a small region compared to the larger CRISP region, and also that it's a protected region and it's closely monitored. And the largest cluster that we identified was furthest away from New York City, up by Otsego Lake, and also near Cooperstown, which is located at the southern end of the lake. And we know that this is also an area that has a biological field station for SUNY Oneonta, which may be a variable of interest when we're doing our analysis. And for the invasive species sightings, we used IMAP Invasives, which is regulated by the New York Natural Heritage Program. And that means that we may not be using other data that also could show certain invasive species, but this is the reliable state-sponsored database that we're looking at. So it's still good data, and we know exactly where these sightings have occurred. So we're sticking with IMAP Invasives, but it doesn't necessarily mean that it's every single invasive species in the region, because we know that this is a huge region and there's likely other invasive species that haven't been detected yet. So the proportional symbol map shows similar results to the kernel density analysis. Again, Otsego Lake up in the north, up north of Oneonta, is the hotspot that we've identified as being the most dense location for invasive species. If you look at some of the HUC boundaries there, if you look to the north, you see one super high compared to HUC boundaries down in the Catskill Park that contain only one, for example. So we'll move on to the core pleth map that showed the same results as well. Also showing that the area around Canada-Rago Lake and Otsego Lake were the highest proportion of invasive species sightings within the overall CRISP region. So our expected results should show the clustering in the northern part of the study area is not random. And this is something we have determined already. Professor did a Moran's eye analysis that showed that the spatial clustering that we identified as being in that northern area is not by chance. And there may be other variables at play, but we do believe that proximity to human population is not the major reason for the clustering that occurred here, but rather recreational behavior, because we know these areas are popular areas for fishing and boating, and those are also activities that we know to be dangerous for transmitting invasive species, which is why the New York State Stewardship Program exists in the first place. So some of those recreational activities, kayaking, fishing, boating in general. So in policy terms, we expect that this probably supports stewards in or near present locations that they do boat checks and like cleaning and regulating the waters, just because there's such a high density in those areas. Some work to complete. The spatial autocorrelation is going to attach statistics to the clustering shown in the heat map. That's something that we already confirmed exists with the Moran's eye, but we're also expecting to do a regression analysis with all of our variables, the dependent variable being the reported counts of invasive species segmented by those hydraulic unit boundaries that we've honed in on, and all of our independent variables, which we have the data for, and we're about to do the regression analysis on. So we have boat launch sites, we have trout fishing sites, New York DEC trails, proximity to roads, proximity to interstate exits, population, number of residences, information available from the newly released census data, land cover composition, lake depth, and finally turbidity. So these are all variables that we expect to run a linear regression analysis on to determine the relationship they may have with the sightings of invasive species as per the IMAP invasives data. And another thing that isn't in the scope of the grant, but we learned a lot about during our literature review, is remote sensing. So we're going to include an example that we thought was applicable and could be a model for future studies in this area. Remote sensing involves assigning pixels to range values so that similar areas are grouped together. So a satellite or a drone may take images of a certain area, and based on a spectrum of ranges of values that come back from the data included in the image, certain values are assigned to each pixel, and those are grouped together. So if you had a map per se with two different colors, the multispectral imagery would show that and pick up that those are two different colors, and one may be invasive and the other is not. And we'll include some pictures. So these are called training sample data in ArcGIS Pro. So what we looked at was aquatic vegetation in South Africa. This process involves high resolution data, and ideally the data would be continuously accessible and updated through a server for daily monitoring. But right now the process is more appropriate for national level rather than regional analysis. But we do expect that remote sensing studies are going to become more affordable in coming years. So this could definitely be something within the scope of a future grant. And these on the right are the images taken from the lake in South Africa where they were looking for water hyacinth. So in the top model, you can see some areas of the lake, mainly the northern area and that cove up there, are hotspots for water hyacinth, which is a dense matted plant, aquatic invasive. And it may not be similar to what we exactly experience here, but it could be a model for future modeling for species that demonstrate those characteristics as well. And the various indices are used to measure or used to predict the location of the water hyacinth, and there's different indices because different models will not always predict the same exact thing. So the more sensitive models may pick up spots that are not necessarily invasive species, and moderate models or low models may pick up areas that are invasive species inhabited but appear not to be. So this is also something that we're not necessarily doing, but is interesting nonetheless and could be applied in the future. And that would wrap up where we're at now. We just have to do the linear regression analysis and write the paper, and hopefully the linear regression can provide some answers about what variables have the biggest influence on where these invasive species sightings are, and allow us to guide the New York State Stewardship Program to appropriate locations for monitoring the waterways. Thank you.
Yeah, thank you so much. That was really amazing. And especially thank you, Hani, for already starting to answer questions that are coming in. So if you ask your questions right into the Q&A section, Hani and TJ will be able to write you a response. And I think we're just going to turn it right over to Marissa Cordell to make sure you have enough time to share your work with us. So go ahead.
Yeah, let me go share my screen. There we go. Sorry about this. Okay, and okay, so my project this summer, we wanted to evaluate the single and combined effects of co-occurring stressors of white-tailed deer, invasive earthworms, and invasive plants. And so oftentimes multiple stressors will occur within the same ecosystem. And when this happens, it can be hard to distinguish what the real driver of community changes are. And in addition, multiple stressors can have synergistic effects, sort of adding together, driving community composition. And so over here, I've included photos of the invasive earthworm and the invasive plant that we studied, and deer. And so oftentimes invasive plants are high priority for managers. Invasive plants, they're pretty visible, and removal is generally the strategy used to deal with them. They can have many effects on forests. They compete for resources and space with native species, and can cause declines in performance and survival of native species. Microstegia viminium, also known as stiltgrass, is an invasive C4 grass that has become widespread in northeastern forests and was the invasive species that we used in our study. So jumping worms are invasive earthworms that have been introduced from Asia. Invasive earthworms can also have many negative impacts on forests. They can change the soil structure. They can affect water percolation throughout the soil, and also affect nutrient dynamics within the soil. This photo down here on the bottom, on the left side of this photo, this was taken in June of 2016. And over here on the right, this photo was taken in August of 2016. And it really just shows how much of a difference these jumping worms can make when they're present in an area. I mean, they just completely decimated all of the leaf litter in the area and completely changed the structure of the forest there. Currently, we have 3 species that are widely distributed throughout areas in the Mid-Atlantic region and in New York State. And so white-tailed deer populations also have been expanding due to decreases in predators and increases in agriculture. Deer often will preferentially browse on native species and will avoid non-native species, which can positively impact invasive species. And we've seen this within stiltgrass as well. And so this summer, our objective was to assess the single and combined threats of white-tailed deer, jumping worms, and stiltgrass on native understory plants. And in addition, our research also aims to raise awareness about invasive earthworms to local managers and communities, as this invasion has really only just begun in places and many people don't know about them yet. And so our hypothesis for this study is that these stressors of stiltgrass, deer browsing, and jumping worm abundance all on their own can cause decreased performance within two native plants. However, when these occur within the same ecosystem, they can have synergistic effects, causing a greater decrease in performance. And we think what's really driving this decrease in performance is going to be deer browsing and jumping worm abundance. And so what we did is we selected 7 sites throughout the Catskills region. You can see the 4 red stars here are where our sites were located. All of these sites already had deer exclusion fences previously built, and each site had varying amounts of jumping worms and stiltgrass. And so at each site, we had 2 plots. We had an open plot where there was no fence and deer browsing would be occurring in this area. And we had a closed plot where there was a deer exclusion fence where deer cannot enter and would not be browsing. And we planted 20 seedlings of each of the 4 forest species that you see over here on your right. We used up at the top left here, bottle brush grass. On the top right, red oak. In the middle here was Zig Zag Goldenrod. On the bottom is lady fern. And so I want to point out that the 3 species, these 3 pictures right here, these are pretty mature versions of these seedlings, and the ones that we used were much smaller and like very small species. And in the next slide, I'm going to show you a photo of the actual seedlings that we used, and you can see how small these seedlings actually were. And so this is a picture of our seedlings. Up here at the top, we have our red oak. Right here, this little guy right here is our bottle brush grass. Down here at the bottom is the Zig Zag Goldenrod. And over here on the left is the lady fern. And so obviously these are much smaller than any of the mature species. And so after we transplanted the seedlings to all the sites, we sampled for jumping worm abundance using mustard sampling. You can see here on the picture on the left. And what you do is you essentially pour a mustard solution over the soil, which irritates the skin of the invertebrates, and they come to the surface, so you can sample for them. And then we measured percent cover of the stiltgrass. And so an example of that is over here on the right. So for this, you just kind of lay down this quadrant here, and you estimate how much of the area inside is covered by stiltgrass. We also installed lysimeters to take water collection so that we could evaluate if changes in water percolation and soil nutrients were occurring. And we also took soil cores to evaluate soil characteristics and changes in soil characteristics. At the end of the summer, we recorded survival of all of the seedlings. And so what we found, these first 2 graphs are just kind of correlational graphs. And so this one on the left, on the y-axis, we have jumping worm abundance, and on the x-axis, we have plot. And so what this graph is telling us is that within the fenced plots, we're seeing higher jumping worm abundance than within the open plots. And then this graph over here on the right has percent cover of the stiltgrass on the left. And along the x-axis, again, we have plot. And so what this graph is telling us here is that within the open plots, we have higher amounts of stilt grass than in the fence plots, which goes along with our expectations of deer having a positive impact and kind of helping the survival of stilt grass. For our single effects, we found that red oak survival decreased significantly in response to increasing jumping worm abundance. And so in the graph along the x-axis, we have jumping worm abundance, and along the y-axis here, we have red oak survival. And so to begin with no jumping worms present, we have about 95% survival of the red oaks. And as jumping worm abundance increases, the survival of red oak decreases to about 30%. And I really just want to emphasize how astonishing this result is, because these seedlings were only monitored for a couple of months, and to see such a huge decrease in survival in response to jumping worms is just really quite astonishing. And so for our interactive effects, we saw that there was interactive effects between stilt grass and fencing and no fencing. And so this graph, along the x-axis, we have percent cover of stilt grass. And then along the y-axis, we have increasing amounts of bottlebrush grass survival. And so here we have on the left side, when the percent cover of stilt grass is very low, there is really no difference, no major difference between the survival of the bottlebrush grass. But within the fence plots, denoted here by the black line, we don't see a decrease in survival in response to the cover of stilt grass. So what this is telling us is that in combination, deer presence and stilt grass are what's causing a decline in the bottlebrush grass. And so this blue line is really showing that. So within the open plots where deer are present, we're seeing a decrease in survival of bottlebrush grass as the percent cover of stilt grass increases. And so again, we got more interacting effects of jumping worms and stilt grass on Zig Zag Goldenrod survival. And so this graph is showing here jumping worm abundance along the bottom, and then Zig Zag Goldenrod on the y-axis. And these three lines are representative of three different amounts of cover of stilt grass. And so this blue line represents about 10% cover of a relatively low amount of stilt grass. And so this blue line is saying that, Zig Zag Goldenrod can survive jumping worm abundance even at high levels when stilt grass is not present. But as we increase the amount of stilt grass in the area in conjunction with jumping worm abundance, we see a large decrease in the survival of the goldenrod. So this green line here represents about 50% cover in stilt grass, and we go down to about 50% survival. And then with 100% cover of stilt grass, we go down to just very low levels of survival of the Zig Zag Goldenrod. And so really, these invasive species are having an effect on the survival of our native species. And so while this project was just for the summer, we do have permits to continue monitoring, and we will continue to monitor our seedlings for performance and survival throughout, at the very least, this next field season. Currently, we are working to assess the species distribution of jumping worms across all of the sites. In addition, the soil core data that we have, the soil cores have been processed. The data is just waiting to be analyzed currently, which will be happening in the next few weeks. The data that we've gathered from the lysimeters can help us identify changes within water flow and nutrients in the soil. And so basically some major conclusions from this project. Clearly, these invasive species are influencing the survival of these native species. And our results indicate that species response is specific to a stressor. So, right, so how we saw with the red oak, it was jumping worm abundance that caused decreases in survival and not, cover of stiltgrass or deer browsing. But with the bottle brush grass, it was stiltgrass and deer browsing that caused a decrease. And so also we found that these stressors are having synergistic effects on native species survival as well. And I would like to bring up that we did transplant the lady ferns, but we did see no significant effects on any of the lady ferns. Previous research indicates that ferns, just for some reason, can withstand stressors for longer than other species. And maybe it's just that they have like a bigger threshold to be able to deal with this. And coming back to our hypothesis, so we did see, decreases in plant performance due to single effects, but we did also see synergistic effects on native plant performance as well. And so this research is really great. I think the data that we collected is, really important. And this data is necessary for us to be able to make informed decisions on invasive species management. Without this data, it would be kind of useless to try and help the red oak and manage for the red oak by removing stiltgrass when the data is showing that it's jumping worm abundance that's really causing declines in that species. And so research like this can help us identify what the real drivers of declines in native species are and can inform managers of the most effective solution. And also, this data can also be implemented, this data collection can also be implemented pretty easily. It's a relatively cheap form of research, and there isn't also, a ton of expertise needed to be able to do this, to transplant seedlings and then monitor them and collect some data. Also, I would just like to acknowledge the Carey Institute for funding and the summer fellowship. I'd also like to acknowledge Tim McKay and Annis Dobson as collaborators on this project from Colgate and Yale. And of course, my lovely advisors, Andrea Davalos and Laura Eierman, just for their continued support throughout this whole project. And with that, are there any questions?
Thank you so much, Marissa. That was really amazing. And yes, you have some questions in the chat. And since we have time, I'm just going to read them out and let you answer them live.
Yeah.
So the first one, Tristan Tabor asks, what was your N for this project? Sorry if I missed how many plots of each type you said were in the experiment.
Okay, so we had 7 sites and each site had 2 plots. So we had a total of 14 plots. And across all of the plots we transplanted 280 seedlings of each species.
Okay, Gary Lovett asks, could you tell why the jumping worms decrease survival of oaks? Was there a decrease in root mass or some other observable symptom?
No, honestly, when we went back to record like species survival, we really just looked to see if there was a presence or absence of the red oaks. And so as we go back throughout like the next year or so, we want to also measure for performance, but so we took measurements at the beginning of the summer, and you can see in the one picture, I don't know if I can go back. That's weird. The one picture, I'm measuring the size of the red oak, but just within the scope of like a 3-month project, there wasn't going to be enough growth for us to assess performance of the plants. So we just stuck with survival for this.
Okay, and then do you know if there are allelopathic qualities of stiltgrass?
That I am not sure of. That is a great question, and I do not have the answer to that question. I'm sorry.
No worries. So Jamie Deppin asks, you may have explained this already, but why did you decide to test these specific native species that you chose?
So we chose these 4 different species. Let me go back. I can bring the picture of all of them up again. So we chose these 4 different species. Originally, we had thought about doing just specifically woody species, but we chose kind of like a wide range of different types of species. So we have the red oak as a woody species, and then we have the lady fern as a fern, and like a grass as well. So I think it gives us kind of a nice range of to see if there were different effects on different kinds of species.
From Joshua Arno, we have, how do you manage or remove jumping worms? And also, why does the absence of deer reduce the levels of stiltgrass?
Okay, so first I'll answer the managing for the jumping worms. So, right, it would depend on the level of invasion in an area. So, you know, is this a brand new invasion where there's not that many jumping worms, and or is this a pretty severe invasion where there's a lot? And I think at the level of there being a pretty severe invasion, at that point, there's not much that you can do. It would be kind of hard to do any sort of removal, and any sort of like something to put in the soil to kill them would probably not be the best for other species. So currently, I'm not sure if there's too many things that we know that we can do to manage for these, except for, you know, if it's an interactive effect that we're seeing, maybe we can manage for something else. So, right, how we saw with the jumping worms and the stiltgrass, the Zig Zag Goldenrod can survive pretty well with jumping worm abundance, but you know, maybe we can do something to remove the stiltgrass in the area, and so the Zig Goldenrod will survive in that area. And then, I'm sorry, what was the second question again there?
Oh. It was, why does the absence of deer reduce the levels of stiltgrass?
So actually, the levels of deer, so it's either that deer are browsing on these species, but we really don't think that that's what's happening here. As I said, like, these fences were already constructed, and so within the fenced plots, there's already probably some community changes happening within the species there. And so we think that it's changes in like community structure that are really driving at least this result that we saw.
How large were your exposures?
So it actually depended per site. Some of the exposures were quite large, whereas some of them were, you know, I don't know, the size of, you know, a large room, I guess. I don't have the exact measurements, but some of them were very large, and then some of them were, you know, more moderately sized. Are there any other questions?
Well, great. Kara, do you have anything else to add, or should I thank people and sum up? Okay. My gosh, we lost Kara. Oh, there she is. She's coming back on. There's some notes in the chat. Just wanted to, Andrea, the mentor. Worms have good years when populations are large and years when populations are low. This is true of many things. I really enjoyed these talks. Thank you, Marissa, Hanny, and TJ. They were really excellent. They brought out some of the most important points about invasives. Not all non-natives are invasive. Not all invasives are non-natives, right? That's a really important point for people to realize. I think the other point that Marissa just was addressing, that, you know, once either non-natives or natives become invasive, it's awfully hard to manage them, right? You can try, there are biological controls. As Andrea said, there are good years and bad years. You know, we know that with ticks, 18 months after you have a lot of acorns, you get a lot of ticks, because in the middle there, you get a lot of mice, and that's what's driving tick populations. We know that carbon dioxide and temperature are increasing the invasive qualities of one of our favorite plants, poison ivy, which is a native. There are lots of stories here. This is why Gary Lovett, founder of the Catskill Science Collaborative, has spent much of the last 6 years, almost 7 years actually, working on summarizing what we know about forest pests and pathogens, particularly invasive pests and pathogens, and particularly the non-native ones, and where they come from overseas, how they get here, and how we can change policy and management through something we're calling Tree Smart Trade. So if you Google Cary Institute Tree Smart Trade, you can learn more about that. But fundamentally, the study of invasives has been at the heart of Cary Institute's work, whether it's forest invasives or zebra mussels in the Hudson River for many, many decades. And it is an area of great importance as global trade continues to shift things around, as climate change continues to make things more attractive to invasives, and as we do lots of things to disturb habitats and make them also great habitat for invasive species. So thank you to our Catskill Science Research Fellows for remarkable work. Thank you to the mentors for such remarkable mentoring. And thank you to the 37 attendees for coming and learning more. Please go to the Cary Institute website, Catskill Science Collaborative, to learn more about the collaborative, sign up for getting notices on upcoming events. And we really appreciate your support. One last thing, if you like this, let your friends at DEC know you liked it, just because they are our core funders, and we're very grateful to them for the support we get from the Environmental Protection Fund. And they realize more than anyone else, except maybe tied with DEP, the importance of good science for management, and how science doesn't tell us necessarily what to do, but it can give us options, and it allows us to monitor and look at the impact of our actions and come up with new ideas. So thank you for being here, and thank you all again for such great presentations. We look forward to seeing you for our next Catskill Science Collaborative Outreach webinar. So anyone, John, Kara, anything to add?
No, that's great. Thank you so
presentation.
much, Josh.
Okay.
Thank you.
All right. Thank you all, and have a great weekend.
Bye. Have a
Bye.
good day.